So we're going to discuss the Enterobacteriaceae with the macaque, the oxidase in the gram-negative rods. So the first thing to do, the first thing we discuss is the family of Enterobacteriaceae. There are members are free living in nature and also found in the intestines of man and animals. There are enterics, so clinically relevant members are opportunistic pathogens, which means that they are coliforms, Escherichia, Klebsiella, Enterobacter, Serratia, Centrobacter, Proteus, Providencia, Morganella, which they are the common cause of pneumonia, meningitis, septicemia, UTI, and wound infection. And we have the other one, which is the overt pathogens, which cause disease in gastrointestinal tract of man, like Salmonella, Shigella, and Yersinia. They can only found in man at times of infection. Fecal oral routes, except Yersinia pestis. So their biochemical and morphologic properties are that they are gram-negative, they are Coccobacilli, they are oxidase negative, which except for Pseudomonas species. So they grow well on macaque agar and on EMB. So the EMB means the eosinophil methylene blue. There are ferment glucose, which they are acid or acid and gas. They are either anaerobic, anaerogenic, or aerogenic. They are facultatively anaerobic, and there are motile, which is their peritrichous, except for Tatumella petitius, or non-motile, they're like Shigella and Klebsiella. So they grow well on routine like blood agar, like citrate agar, BHIB, endothio, and they are asporogenous. So yeah, so they reduce nitrate to nitrite, except for Pleurohabdus, Xenorhabdus, and some strains of Enterobacter and Yersinia. They are catalyst positive, except for Shigella dysentery type 1 and a species of Xenohabdus. So okay. So there are antigenic structure. Let's start with the somatic antigen, which they are OAJ. There are polysaccharide chains of lipopolysaccharide or endotoxin components of outer membrane. They are heat stable. They identified with specific O antiserum. And we have the next thing is the capsular antigen, which there are KAJ and VIAJ, which cover the OAJ. They are heat label. They're heating in boiling water bath for 10 minutes. And we have the flagellar antigen, HAJ, found in the chon that makes up the flagella. They are heat label, can be destroyed with ethanol. So their serotyping should be done using contains from a non-sugar containing medium such as blood agar to prevent false positive results. So in the media for isolation, so let's discuss first the differential mildly selective. They are carbohydrates, pH indicator, chemicals, inhibit gram-positive bacteria. So we have the macaque agar, which there are lactose, eusyl methylene blue agar, which there are lactose and sucrose, Lipson, deoxycholate agar, and their lactose. So there are two groups of it, which there are lactose fermenters, which are colored colonies, most coliforms, and there are non-lactose fermenters, which are colorless colonies, most enteropathogens. Okay. Now we're going to talk about the differential moderately selective. There are carbohydrates, pH indicators, chemicals, inhibit gram-positive bacteria, and most coliform for isolation of Salmonella and Shigella. So the Salmonella Shigella agar or SSA, there are colorless colonies. H2S producers show black centers. There are xylose, lysine, deoxy, and that's the description of the Salmonella Shigella agar. So let's talk about the xylose, lysine, deoxycholate agar or XLD. There are Shigella, which is colorless. We have the Salmonella, which are red colonies. We have H2S producers show black centers. And that's the description of the xylose, lysine, deoxycholate agar. Then we have the deoxycholate citrate agar or DCA. There are colorless colonies. So let's go with the Hector and enteric agar or HE. They are bluish-green colonies which they are H2S producers. Shows black centers. So yeah. So now let's talk about the highly selective media. They promote growth of a specific bacterium or a group of bacteria. So first we have the bismuth sulfite agar, BSA, which there are Salmonella typhi. They are jet black colonies. Then we have the brilliant green agar or BJA. There are Salmonella species, white to pink or red colonies surrounded by a bright halo. Then we have the Cephsoludin ergasan novobiusin agar or CIN, which the Iricinia enterocolitica. There are bullseye colonies, which are dark red centers with translucent borders. Then we have the Mankaki sorbitol agar or Maxor or SMAC. They differentiate E. coli 0157H7, which does not ferment sorbitol from other E. coli strains which ferment sorbitols. Then we have the Chromagar salmonella, which there are Salmonella species produce mauve colored colonies. Now let's go with the enrichment media and technique. These are for stool culture to isolate enteropathogens. Stool or rectal swab is inoculated first into enrichment media before on selected med. So we have the selenite F broth. There are Salmonella typhi and Shingilla species. We have the tetrathionate broth. These are the Salmonella other than Salmonella typhi. Then we have the gram negative broth or GN broth. These are Salmonella and Shingilla. We have the cold enrichment technique. These are the Yersinia enterocolitica, placing specimen into 5 ml phosphate buffer and refrigerated for 3 weeks. So SC are the SC onto CIN med every 4 days until CHA colonies are isolated. So let's talk about the biochemical test. We have the TSI reaction, which detects 3 primary characteristics. The ability to ferment glucose, lactose, and sucrose, which resulted to acid. Then their ability to produce gas like H2 and carbon dioxide. And their ability to produce H2S or cysteinase. Their medium is triple sugar iron agar, which it contains 1% lactose, 1% sucrose, 0.1% glucose, and peptone. Then we have the phenol red indicator. Then there are ferrous sulfate and sodium thiosulfate. They are prepared in a test tube with a slant and slope and a butt covered with a loose cover. Ideal to use cotton plugs or loose cap. So the method of inoculation is you need a wire needle, make a line on slant, stab the butt until a few millimeters from the bottom, then streak across the line on the slant surface. Incubate at 35 degrees in ambient air for 18 to 24 hours. Early or delayed reading of results may lead to invalid interpretation. So, okay. So, we're going to discuss the reading and interpretation of the TSI. The reading on the left side is for the slant, while the reading on the right side is for the butt. So, alkaline over acid, only glucose is fermented on this result, which slant is the pink, butt is the yellow. If acid over acid, the glucose, lactose, and our sucrose are fermented. So, slant and butt are both yellow. If it is alkaline and over alkaline, glucose, lactose, and sucrose are not fermented, which peptone is utilized aerobically and anaerobically. Their slant and butt is pink. If it is alkaline and over no change, glucose, lactose, and sucrose are not fermented. Peptone is utilized aerobically only in the slant, not in the butt. So the slant is the pink while the butt is the orange. The color of the medium is retained, that's why it is orange. So the gas production is H2 or carbon dioxide. If this is produced, crack or bubbles in the butt are observed. Add G in the butt or encircle butt reaction. Like this example, A over A, Ag, and A over A or gas, like that. So H2S production, there are blackening in the butt, involves a two-step reaction. the bacterium or syntenase plus sodium thiosulfate that increases the sodium thiosulfate, which increases the H2S. Then if it is H2S plus ferrous sulfate, it decreases the FES. Add with H2S in the bath reaction. For an example, acid over acid with H2S or A over AJ with H2S. So that's the process of it. So the summary of TSI interpretation. So the alkaline are alkaline over acid. In the slant, they're pink. The bath is yellow. Then their only positive is the glucose. Peptone is non-active. There's no activity. If acid over acid, then slant and bath are both yellow. Then all glucose, lactose, and sucrose are positive. And the peptone is no activity. If it is alkaline over alkaline, then slant and bath are all pink. And though glucose, lactose, and sucrose are positive, no means no. But the peptone is aerobically or anaerobically. In the alkaline over no change, pink is the slant and the orange is the bath, which all the sugars are negative. Their peptone is aerobically. So complete the explanation. TSI, you have to wait 6 to 8 hours over acid, and if it is LF over equals over A, then alkaline over A it results to alkaline over A. If it is 80 to 24 hours, then it becomes to 48 to 72 hours of observation. Then if it is NLF is equal to alkaline over A, then the alkaline over alkaline is the results. So I don't think we don't need this, but I think we need this. So TSI and LIA reaction used to screen the enteropathogenic Enterobacteriaceae and Aeromonas vibrio species. So TSI reaction, if K over A with the H2S production or K over A with gas production or K over A with H2S. So the LIA reactions is K over K or K over NC H2S. Then the possible identification would be the Salmonella serotypes and the Edwardsiella species. If the TSI reaction is K over A with an H2S production, then the LIA reaction is K over K or the same thing with the previous one. Then the possible identification would be Salmonella serotype, which are rare. If it is K over A with the gas production, the K over K or K over no change, the Salmonella serotypes are rare. It resulted in the possible identification of Salmonella serotypes, which are rare also. If it is K over A with H2S production, then the LIA reaction is K over K or K over NC H2S. Then the Salmonella serotype is the possible identification. The Salmonella serotype type P are rare, which they are rare actually. So if K over A with a gas production, then the LIA reaction is K over A with the H2S production, which their possible identification would be Salmonella serotype paratype PA, usually HS. If K over A with a gas production in the TSI reaction, then the LIA reaction is K over A or A over A, which the possible identification for it is the Escherichia species, Salmonella serotype paratype A, Shingilla flexneri 6 or they are uncommon, and Aronoma species they are oxidase positive. So yeah, in the TSI reaction naman, if K over A naman, so the LIA reaction will be K over K or K over no change, which their possible identification would be Leishmaniasis species or oxidase positive, Salmonella serotype type A are rare, which they are Vibrio species or oxidase positive. If the TSI reaction is K over A, then the LIA reaction is K over A or A over A, which there are the possible identification for it is the Escherichia species, the Shingilla group AD, and the Yersinia species. If the TSI reaction is A over A with a gas production of H2S also, then the LIA reaction will be K over K or K over no change with a H2S production. Then the Salmonella serotypes is the possible identification. If the A over A naman, the LIA reaction is K over A or A over A, Then the possible identification would be Escherichia species, Yersinia species, Ironoma species, which they are oxidase positive, and the Vibrio cholerae, which they are rare oxidase positive. If the TSI reaction is A over A, then the LIA reaction is K over K or K over no change, then Vibrio species is the possible identification with the oxidase positive. So in acts of review, so use of triple sugar iron agar useful for detection of Yersinia enterocolytica. Their composition is 10 parts of lactose, 10 parts of sucrose, and 1 part of glucose. They are H2S indicator, which they are sodium thiosulfate and ferrous sulfate. They are pH indicator, phenol red. Function, they are detection of fermentation, gas production, and H2S production. They are blackening in the back of tube. Then the TSI reaction will be enoch by stab in strict. So this would be it. So A over A is equal to or more sugar fermented, which these are the Escherichia, Klebsiella, Enterobacter, and Y in Yersinia enterocolytica. If A over AJ, there are 2 or more sugars fermented with gas. So Escherichia coli, Klebsiella, Enterobacter, Sriracha, that's their pathological identification maybe. If A over AJ plus, there are 2 or more sugars fermented with gas and H2S production, which this could either be Citrobacter freondiae. If K over A naman, the glucose only fermented, so Shingilla, some Hafnia, and some Yersinia pestis are identified. If K over AJ, their glucose only fermented with gas, so Morganella, Providencia, Hafnia, and Citrobacter diversus, that's their one. If K over AJ plus, then glucose only fermented with gas with H2S production, there are Salmonella. So if K over A naman plus, the glucose only fermented and H2S production, so this can either be Proteus and Salmonella. So if K over K naman, no sugar fermented, non-fermenters, not enteric, so Pseudomonas acinobacter. So if no B and B, so the Kia-Kingler iron agar has 2 sugars only, which is the glucose and the lactose only. So let's start with that's kind of long actually. Now we start with the idle production, which is their reagent is, which their finding is the tryptopanase. So their medium is trypton broth. Their reagent is Kovacs reagent or Ehrlich's reagent, which their positive is red or purple ring on top of medium, while the negative is the yellow ring. So the procedure of it is they suspend 1 loophole of the test organism and into the broth, then incubate at 35 degrees for 18 to 24 hours. After incubation, 5 drops of Kovacs reagent or Ehrlich's reagent into the broth. The interpretation results. So the Kovacs reagent, they are positive, red ring on top of medium. If they are negative, they are yellow ring on top of medium. In the Ehrlich's reagent, the positive, they are purple ring on top of medium, while the negative naman is yellow ring on top of medium. That's the... So let's discuss the methyl red test and Vogel's precursor test. These are MR tests, which they are acetate and formate, strong acid. Their medium are MRVP broth. So the reagent is 5 drops of methyl red indicator. The positive of the MR test would be that red medium is pH to 4.5 or less. So their negative naman is that the yellow medium has a pH of more than 5.6. So the VR test, let's talk about the VR test. There are acetoin or acetamethylcarbinol. Their medium is MRVP broth. Their reagent is 6 drops of alpha-naphtol plus 2 drops of 40% of KOH. So their positive appearance would be red ring is formed, then their negative is no red ring produced. So let's talk about the citrate utilization test. Their findings is their citratase. So the medium is the Simmons citrate agar slant or the SCA. there are sodium citrate, ammonium salts, and bromthiamyl blue. So their positive appearance would be the blue, medium, and growth on slant, which is the NaHCO3 and NH3. So their negative naman is green, medium, and no growth on slant. So the procedure of this would be get one loophole of the test organism, inoculate the slant by simple streaking, or you can use a wire needle, get the inoculum and streak the slant in the simple streaking. So the positive naman, the appearance of the positive is Foxian blue, medium in growth on slant, NaHCO3 and NH3 produces alkaline reaction, hence blue color. So the negative naman is green, medium, and no growth on slant. So in the IMBC test, so let's talk about the IMBC test. The IMBC stands for the idle test, methyl red and Voges precursor test, and citrate test, citrate utilization. So let's start with the idle test. This is the detection of enzyme tryptophanase. Their medium is tryptone broth, SIM. Their indicator is Kovacs or Ehrlich's reagent by the p-dimethyl minobenzylaldehyde. So their results is positive, which it is red color and red ring. Their rapid indole spot test is reagent dimenocinnamaldehyde, and their positive naman is blue. So useful test to differentiate P. vulgaris from P. mirabilis. So methyl red and Voges precursor test detects the pathway of glucose metabolism. Medium MRVP broth contains glucose. Methyl red detects mixed acid, utilize the mixed acid path. The pH 4.4 or less. So the indicator reagent is the methyl red. Expected results are the red color. So Voges precursor detects acetoin, utilize the butylene glycol path. So the pH is 5.5 or more. So the reagent is alpha-naptol and KOH or the Barrett's. The alpha-naptol in 40% KOH and creatine covalence. So their expected results is red color. So in the citrate utilization naman, it determines if the organism use citrates as sole source of carbon. Medium Simon citrate agar slants contains citrate source of carbon. Their pH indicator naman is bromthiamyl blue, which their positive result naman is growth, colony change color from green to blue. So, yeah. So, in summary of the biochemical reaction, let's skip that one. So, because it's too long and it's not that important. So, let's go to the motility test. So, there are growing bacteria in semisolid made in bat tubes. So, mediums, sa SIM, it is acronym for sulfide, idle, and motility. They are stub but until a few millimeters from bottom. So their motile is hazy medium, which growth spreading into medium. So their non-motile are clear medium, only in stub line and top of medium. Their H2S production naman is blackening of medium. So their idle production naman is red ring after addition of reagent. So yeah. So in the urease test naman, it splits urea into NH3 and CO2, requires heavy inoculum, 5 loopholes of the organism colony. So their medium is urea broth, phenol red indicator, then their positive is dark pink medium. So the positive naman sa in appearance is that they are dark pink or fuchsia. It has an enzyme urease splitting NH3 and CO2, producing an alkaline pH in the medium. While their negative naman is no change in color, still orange, but retains the buff color of the medium. And that's the description of it. Now let's go to the nitrate reduction test, which the nitratase or nitrate reductase is. Reduce nitrate to nitrite. So that's the goal here. So their medium is nitrate agar slant. The reagent is 2 drops of sulfonylate acid and 2 drops of alpha-naphthalene. Their positive is red azo compound, which there are p-sulfobenzene azo alpha-naphthylamine. They are using a wire loop or wire needle. Get the inoculum and do simple streaking across the slant. Then incubate. So their reagent is added after incubation. So 2 drops of sulfonylate acid followed by 2 drops of alpha-naphthylamine. So their positive naman is the formation of a red azo compound on the slant. So their p-sulfobenzene azo is alpha-naphthylamine. So yeah. So let's go with the phenylalanine deaminase test. They are the deaminates phenylalanine to phenylpyruvic acid. Their medium is phenylalanine agar slant. The reagent is 5 drops of ferric chloride, and their positive is the green slant, which is the Proteus, Morganella, and Providencia. So the procedure of it is using wire loop or wire needle, get the inoculum and do simple streaking across the slant, then incubate. So the positive result may be green slant, while the negative results be no green slant formation. There is a yellow color on the slant because of the dark yellow color of the ferric chloride. The control C is none. So another medium in the market is the phenylalanine agar, wherein ferric chloride is already incorporated with will yield different colors reaction. Their positive is brown slant. So yeah. Now let's talk about the phenylalanine deaminase. It is an enzyme that removes amino group from an amino acid. Their medium is phenylalanine agar with 10% ferric chloride. Their result is green. So yeah. Then we have the ONPG test, or the O-nitrophenyl beta D-galactopyranoside, which detect beta galactosidase. So the two enzyme in lactose fermentation, which is the permease, which allows lactose into bacterial cell, and the beta galactosidase, which splits lactose into glucose and galactose. Their SLF is a lactose permease. So their medium is ONPG broth. It could be 3 to 6 hours incubation, while their positive is yellow medium, which it has a O-nitrophenol, and their negative is colorless medium. The ONPG positive is a lactose fermenters, which there are positive. If it is positive, the appearance would be yellow medium O-nitrophenol. If beta galactosidase is present, it will hydrolyze ONPG and liberates O-nitrophenol, a yellow compound. Hence, the bacterium is a lactose fermenters, negative colorless medium. There are two types of lactose fermenters. We have fast lactose fermenters, those that have both enzyme mentioned above, and we have the slow lactose fermenters, which lacks permease. So ONPG naman is the orthonitrophenyl beta D-galactosinase. It determines lactose fermentation, especially late lactose fermenters. It is the detection of enzyme B beta galactosidase and the substrate ONPG o nitrobeta D-galactokinase. Expected result is yellow coloration within 4 hours of incubation. So let's move on to the lysine decarboxylation and deamination. It detects two enzymes, the lysine decarboxylase and lysine deaminase. Their medium is lysine iron agar or LIA, which their glucose, lysine, and bromcresol purple as pH indicators, which yellow in acidic medium and purple in alkaline medium. It is prepared with slant and butt, just like TSI, but it has a shorter slant and a deeper butt compared to TSI. The aniculated LIA has a purple color throughout the medium. With wire needle, get inoculum and stab the butt twice all the way down to the bottom. Then streak across the slant, then incubate for 6 to 8 hours and read the results. It requires a different method inoculation compared to that of TSI. Since the medium has glucose, the enterics will ferment glucose. After incubation, the medium, which is originally purple, will become yellow because of the acid of the glucose. However, this is not the same case with those which have the lysine decarboxylase and lysine deamination. So, let's comparison the lysine decarboxylation and lysine deamination. So, where to read? In lysine decarboxylation, you need to read in the butt. It is an anaerobic process. So, if it is positive, then the appearance would be purple, which the enteric acts on lysine and produce cadaverine, which is alkaline. Yellow on incubation, but if cadaverine is produced, the butt will revert to purple. That is why it usually has a light yellow tinge because of the acid from glucose, not bright yellow anymore because of the cadaverine produced. If they are negative naman, bright yellow, because the enteric does not have lysine decarboxylase. So now let's move on to lysine deamination. Where to read in the slant because it is an aerobic process. So their positive is red because it can deaminate lysine and produce alpha ketocarboxylic acid, which is responsible for the red color of the slant. Their negative naman is purple slant. So we have three possible LIA reaction. The lysine deaminase, LI 3 possible. If purple over purple, so you have to look if the lysine decarboxylase is positive. And the slant and bat also are both purple naman. Palamang. Purple over yellow, all of it, lysine deaminase and lysine decarboxylase are negative. And their slant and bat naman is purple and yellow. If red over white naman siya, so the lysine deaminase is the only positive. Observe only among Proteus, Providencia, and Morganella, there's no such reaction as RP. Why? This will explains that the enteric may either have lysine decarboxylase and produce PP, or have lysine deaminase and produce RY, or lack both enzyme and produce PY. However, the enteric organism will never have both lysine decarboxylase and lysine deaminase. Indicates that the LIA can also detect H2S production. However, compared to the TSI, LIA is less sensitive for H2S production. So, moving on. So, to summarize things up, use of lysine iron agar, it is the detection of lysine deamination and lysine decarboxylation. Detects H2S production, but results are viable reaction. Composition of glucose lysine, slant where deamination happens, aerobic. The pH indicator naman is Bromcresol purple, which the bat where decarboxylation happens, anaerobic. And the H2S indicator naman is the ferric ammonium citrate, which the production of alkaline product is the cadaverine. So, for an example, if K over K, P over P, the LDC naman, if X Klebsiella is the initial yellow because of glucose fermentation, it becomes purple because of cadaverine LDC positive. If R over A naman or R over Y, they are deaminase positive because in example, its Proteus turns to yellow because it's not LDC deaminase, then it turns to red slant. If K over K naman plus is for P over P with H2S, their CDC and H2S production, example Salmonella. If K over A naman, their P over Y, LDC is negative and deaminase is negative. For an example to beat is the Shingilla, LDC negative, Dim negative, and glucose negative. If K over A naman, P over Y with H2S production, then the deaminase is negative with H2S. One, like that. So, let's go with the ornithine decarboxylation. Their medium is motility, indole, and ornithine. There are semi-solid medium. The glucose, ornithine, bromcresol, purple, they are deep butt tubes. So the positive is purple butt. Putrescine, they're slight yellow tinge. So yeah. And their negative naman is bright yellow butt with purple rim. So their motile is hazy medium. Their non-motile naman is clear medium. Growth in stab line. With a wire needle, get the inoculum and stab the butt once all the way down to the bottom. That's the procedure. Then inoculate. During incubation, glucose is utilized producing acid. The MIO will turn into yellow. So that's the procedure naman. So we have the positive naman, which is the purple butt, which is the putrescine. Those which have the ornithine decarboxylase will act on ornithine and produce putrescine, which is an alkaline product, reverting the butt into a purple color. There is a slight yellow tinge because of the acid from the glucose, and that's the positive. Now, towards the negative naman, they are bright yellow butt with a thin, narrow purple rib at the top of the medium. The motile naman, if the medium is hazy, then it is motile. If non-motile naman, growth confines only in the line of stab and on the top of the medium while the surrounding is clear. So, ornithine decarboxylation can detect three properties of the enteric. Their motility, since it is semi-solid, their indole if you have Kovacs reagent after incubation, and their ornithine decarboxylation. So, decarboxylase. Let's do that. Okay. So, enzyme that removes the carboxyl group from an amino acid is the ornithine, lysine, and arginine. The lysine decarboxylase are the cardivorine production, while the ornithine is the decarboxylase are the putrescine. The arginine dehydrolase are the citrulline, ornithine, and putrescine. Their medium is decarboxylase medium with bromocresol purple, glucose, and amino acid. Their medium is overlaid with mineral oil. The result is purple. It is useful to differentiate Klebsiella from Enterobacter. So, the last but not the least is the malonate utilization. What is malonate utilization? It detects whether the enteric can utilize malonate as the only source of carbon for metabolism. Medium. Malonate broth, bromthiamol blue as pH indicator, just like SCA. Get one loopful of test organism and suspend it to malonate broth, then incubate. So the positive of it is light blue or deep Prussian blue due to formation of sodium hydroxide produced responsible for the blue color. The enteric can utilize malonate as the only source of carbon for metabolism. So the negative naman is the retain of the green color. So what do you think is another difference between malonate utilization test for citrate test? So how about, we're going to discuss that in the next world. So in theory, the miscellaneous biochemical test for enterics, they use the gelatin liquefaction, which detection of enzyme gelatinase. It is useful test for the members of Ceratis. Their medium is 12% gelatin, result gelatin liquefies. Interpret results after refrigeration. Then we have OF medium, which is the oxidation and fermentation to detect if origin is an oxidizer or fermenters. Their pH indicator naman is bromthiamol blue. Their inoculate is 2 tubes, 1 open and 1 closed tube. Overlay with mineral oil to have anaerobic environment. So yeah. So for an example, if fermenters, if it is the open tube, it is yellow. At the same thing in the closed tube. If it is oxidizer, only open tube is yellow, while the closed tube is blue. If it is the acicarolytic, all of them is blue. So yeah. So we're going to talk about the MAG test. They are substrates for methylumbilyl beta-D-glucuronide. It is the detection of enzyme beta-D-glucuronidase. Positive result is the electric blue fluorescence. Useful presumptive ID test for E. coli except EHEC. Useful also for water analysis testing to check contain of fecal matter. So yeah. So the oxidase tests most are negative except trichomonas. So in summary, we don't know. So, we don't know about the summary anymore. So, let's move on to opportunistic coliforms enteric pathogens. They only produce disease if they enter sterile sites in the body, such as blood, but are usually harmless normal flora in the gastrointestinal tract. Hence, they're called enterics. In the genus Escherichia naman, Escherichia coli is the predominant facultatively anaerobic species in the large intestine of man. Index of fecal contamination in water bacteriology. If it is EMB or eosinophil methyl blue, they are greenish metallic sheen. If it is MAC, they are flat dry pink colonies. If it is in the blood agar, then some are beta hemolytic. There are most strains are motile. Lactose fermenters are aerogenics. The TSI naman is equal to none, while the IMVIC naman is positive, negative, negative. Most common cause of UTI is nosocomial infection and gram-negative bacteria. So there are two groups of pathogenic Escherichia coli. You have the extraintestinal pathogens, which are the uropathogenic E. coli or OPEC. These are the major cause of E. coli associated community acquired UTI. causes cystitis and acute pyelonephritis. Their virulence factor is hemolysin, pili, cytotoxic, necrosing factor is an example of E. coli 06K2H1. The next one naman is the meningitis or sepsis, which is associated with E. coli MNEC, causes neonatal meningitis that results in high morbidity and mortality. Virulence factor is K1 antigen 80%, which the example of it is the E. coli 07K1. Now, we have going to the diarrheagic or intestinal pathogens. We have 5 strains which can cause serious gastrointestinal infection, tract infection. We have the enterotoxigenic E. coli ETEC. We have the enteroinvasive E. coli EEC. We have the enterohemorrhagic E. coli EHEC. We have the enteropathogenic E. coli or EPEC. We have enteroaggregative E. coli or IEC. They are transmitted by contaminated food in water. So, in the ETEC naman, their path mechanism is the enterotoxin, LT and or ST, which their primary site naman is small intestine. Their epidemiology naman is the travelers and childhood diarrhea. While their fever is absent, their stool is copious watery, their blood is absent, at the same time their pus. So the EIEC naman is that the invasion of enterocytes are their path mechanism. Their primary sites is the large intestine, and their epidemiology is uncommon dysentery-like syndrome, which their fever is common, their stool is scantily purulent bloody, their blood is common, at the same time their pus is prominent actually in there. In the EHEC, VTEC, and STEC naman, their path mechanism is verotoxin, Shiga-like toxin. Their primary sites are the large intestine, and their epidemiology naman is the hemorrhagic colitis, hemolytic C uremic syndrome or HAS. Their fever is absent, while their stool is copious bloody. Their blood is prominent, and their pus is absent. In the EPEC naman, their path mechanism is attachment and effacement of enterocytes. Their primary sites are small intestine. Epidemiology is the infantile and childhood diarrhea. Their fever is common, their stool is copious watery, their blood is absent, and their pus is minimal. In the EAEC naman, their path mechanism is adherence to enterocytes or AGGR. The primary sites is unknown, while their epidemiology naman is diarrhea in children like HIV infected and AIDS patients. Their fever naman is occasional, their stool is watery, their blood and pus are both absent. So yeah. So let's talk about the ETEC, which is the childhood and travelers diarrhea. Mild watery diarrhea with abdominal cramps and nausea and headache. So their LTN or XST enterotoxin. Your LT enterotoxin are the heat level at 65 degrees for 30 minutes. They are antigenic. The rabbit ileal loop test is positive in the dilation of loop. Their Chinese hamster ovary cell assay is cell death. They reverse passive latex agglutination test. They are DNA hybridization. And the ST enterotoxin naman, they are heat stable even at 100 degrees Celsius. They are poorly antigenic. Their infant's mouse is assay and DNA hybridization. So in the EIEC naman, they cause bloody diarrhea as a result of direct invasion of the enterocytes in the colon. For an example of it is the E. coli 0112. Their Sereni test is the keratoconjunctivitis in 24 hours, which is that. So in the EHEC naman, EHEC, or they are the verotoxin or cytotoxin which damages vascular endothelial cells, causing hemorrhagic colitis. It can also lead to hemolytic uremic syndrome or HAS. HAS is characterized by hemolytic anemia and low platelets count, often results in kidney failure and death. They also referred to as verotoxin producing E. coli or VTEC, or Shiga-like toxin producing E. coli. Most frequently associated with E. coli is 0157H7 and E. coli 0157NM. So there Mankaki sorbitol so there are Mankaki sorbitol agar, the EHEC, which Maxor or SMAC. We can use the suffix Cifixim tellurate containing Mankaki or CTMAC. We can use the Chromagar 0157, has higher sensitivity and specificity and better diagnostic efficiency than SMAC or MAV4. We have serotyping and we have cytotoxin assay, which there are the Vero cells, African green monkey, kidney cells. EPEC does not produce exotoxin associated with attachment and effacement of the intestinal cell wall, causing attachment and effacement lesion in the colon. An example of it is the E. coli 0111 and 0125. They are serotyping. So the EAEC naman is associated with the aggregative regulator gene, AGGR. They are responsible for cellular adherence. Their stool are typically not bloody and do not contain pus cells. Their inflammation is accompanied by fever and abdominal pain. So how is the setup done in the doing the manual identification of bacteria and how to ID? First, always starts with the plate for characteristics growth of the organism, whether in MAC or EMB or in the BAP. Second, look for the key features of the biochemical reaction. Memorize or familiarize at least 3 the usual biochem tests. I take note is the TSI, IMVIC, usually indole and citrate, the MIO, the LIA, the PAD, the PAF or /PAF, and the sugar fermentations. So let's move on to the genus Klebsiella. There are species that are inhabitants of nasopharynx and gastrointestinal tract. They are cause liver abscess, primary pneumonia, UTI, wound infection, septicemia, and meningitis. The Klebsiella pneumonia is also known as the Friedlander's bacillus. There are EMB or AM or MAC. There are large pink mucoid colonies. There are capsulated, non-motile, urease positive. There are lactose fermenters and they are aerogenic. Their TSI is equal. Their MVIC is or IMVIC is equal to the result may be negative, positive. Their Klebsiella pneumoniae containing K1 capsular clonal complex. The CC23K1 is associated with pyogenic liver abscesses. Their presence of another K1 capsular complex is CC82K1. It is associated with severe pneumonia and septicemia. Those that demonstrate hypervirulent, they are also known as the hypermucovicious phenotype or HVKP, are resistant to bactericidal effects of serum complement and neutrophils. So they are resistant to ampicillin and may demonstrate multi-drug resistant because of plasmid with enzymes such as cambapinimase and cephaloporinase. The Klebsiella oxytoca has a heat-labeled cytotoxin, has been isolated from patient with self-limiting antibiotic-associated hemorrhagic colitis. Then we have the Klebsiella rhinoclerosmatis and Klebsiella ozeanae. There are the agents of chronic infection, which there are rhinoscleroma and atrophic rhinitis ozeana. So that's how it ends. So let's talk about the Klebsiella granulomatis. They are formerly known as the Calimantobacterium granulomatis. They are granuloma inguinale or dunovanosis. There are STDcha by genital ulceration and inflammation. They are difficult to cultivate in vitro. Scraping of lesions, right James's stain. There are Donovan's bodies, which are the bacterial stained as blue rods with polar granules. There are safety pins appearance and large pink capsule with large macrophages. So, in theory, the Klebsiella, the normal habits of the gastrointestinal tract of humans and animals found in environment. There are causes of opportunistic and nosocomial infection, which they are the UTI, pneumonia, and wound infection. The Klebsiella pneumoniae, the common name is the Friedlander's bacillus. Their causes lobar pneumonia, their current jelly-like sputum, UTI, and lower respiratory tract infection. The Klebsiella oxytoca is a similar infection caused by Klebsiella pneumoniae. They are unique biochemical tests which the indole is positive, and that's the Klebsiella. So we going to go to genus Enterobacter. They are small capsules, which they are less mucoid colonies. Their eosinol methylene blue is the fish eye colonies. There are motile, lactose fermenters, and aerogenic. Their TSI is equal with their IMVIC is negative, negative, positive, positive. There are 3 species commonly encountered in the lab: the Enterobacter cloacae, Enterobacter aerogens, and Enterobacter agglomerans. The Enterobacter cloacae is the only positive is in the O decarboxylase, while in the L decarboxylase they are negative. In the Enterobacter aerogens naman, they're all positive. In the Enteroagglomerans, they are all negative. So urease is negative except for species of Enterocloacae and Enteroagglomerans. So let's go to Pantoeagglomerans. There are new designation of Enteroagglomerans. There are yellow pigmented colony lysine decarboxylase, ornithine decarboxylase, and arginine dehydrolase negative. So Enterobacter, it causes non-nosocomial and opportunistic infection. There are UTI, respiratory tract infection, wound infections. There are aerogenic, and there are Enterobacter aerogens. They are negative for urease, Enterobacter cloacae, and Enterobacter georgiae. They are from respiratory specimen urease pus. So Chronobacter sakazaki or Enterobacter sakazaki is the production of yellow pigmented colonies at 25 degrees. So Pantoeagglomerans, they are formerly known as the Enterobacter agglomerans. They are septicemia for fluids contamination healthcare setting. They are triple decarboxylase negative, ODC negative, LDC negative, and ADH negative. So yeah. So we're going to the Chronobacter species. So the Chronobacter sakazaki is a neonatal infection, while the Chronobacter malunaticus is an adult infection. They are formerly classified as Enterobacter sakazaki, while associated with bacteremia, meningitis, and necrotizing colitis. They are yellow pigment best demonstrated at 25 degrees. They are resistant to ampicillin and first and second generation of cephalosporin because of AMPC chromosal beta lactamase. There are Voges Procars test negative, and that's the description of the Chronobacter malunaticus. Then we have the genus Hafnia. There are Hafnia albi. There are Enterobacter hafniae. There are SLF. There are indoles negative. There are cytridis negative. Their urease is negative. Their lysines and ornithine decarboxylase are positive. There are more positive results at 25 degrees.